Okay, so Tatum and I focused our presentation on Chapter 7 regarding linkage and recombination. So, what are linkage and recombination? As a recap from what we're already sort of learning in lecture, linkage involves two DNA segments that are associated and exhibit co-inheritance with each other because they are on the same chromosome and are relatively close together. Recombination involves the alleles that can be sorted into various combinations, but they depend strictly on the parent's genes. And linked genes, which is when the genes are close together, can only recombine after crossing over. So why linkage and recombination? Linkage and recombination are so important because they aid in increasing genetic diversity. Linkage is responsible for keeping the genes on the chromosome together, and recombination allows the actual parts of the chromosome to separate and be crossed with the other chromosome in meiosis.
The paper we were assigned is titled A Correlation of Cytological and Genetical Crossing Over in ZMAs, where they focused on crossing over in corn chromosomes. The focus of the paper was on how chromosomes in the same linkage group were impacted by crossing over. The researchers wanted to see if cytological and genetic crossing over occurred together and had any correlation with one another. And it was evident that the corn with abnormal chromosomes exhibited knobs, which are areas that have an increased gene density, which consequently suppresses recombination. So more specifically, the researchers were looking in a certain strain of maize at the second smallest chromosome, which is chromosome 9, which possesses a knob at the end of the short arm and is seen in preceding generations through their findings. So the researchers identified that through cytological observations that the resulting F1 individuals of a plant possessing the knob at both ends, when crossed with a plant with no arms, only one member of the homologous pair will possess a knob. So when that individual is back-crossed, half the offspring are heterozygous for the knob and half possess no knob at all. And the knob was therefore shown to be a constant feature of the chromosome possessing it. And in this specific strain of maize, interchange has shown to take place between chromosome 8 and 9, but the interchanged pieces were unequal in size. So the arms responded with this by lengthening or shortening in size, and the gametes resulted in an individual characterized by a side-by-side synapsis of homologous parts, which will be shown on the next slide. So the researchers found that the individuals with functioning gametes are the ones that possess either normal capital N, lowercase n, or interchanged uppercase I, lowercase I. So the functional maize will either possess the N, shorter normal knobbed chromosome, or capital I, longer knobbed interchanged chromosome, which is shown on the top two images. Figure 1 on the right side shows some of the various types of chromosomes that the researchers saw in their experiment. For when no crossing over occurred, the gametes would contain either a knobbed interchanged chromosome, which is shown by letter A, or a normal chromosome with no knob, shown by letter D. And when crossing over did occur, the result would be seen as a knobbed normal chromosome, represented by C, or knobless interchanged chromosome, represented by B. So the knobbed chromosomes were shown to carry the genes for colored allurone by C, or shrunken endosperm SH and waxy endosperm WX, and that they lie in the short arm of the knobbed chromosome. So therefore a linkage between the knob and genes is to be expected. And in order to obtain the cytological and genetic crossing over correlation, the plant must be heteromorphic for the knob, gene C, and WX, as well as the interchange. So when pairing the chromosomes heteromorphic in two genes shown to exchange parts at the same time, they exchange genes assigned to these regions. And the image below is just a representation of the crossing over of these genes that will represent those genetic crossing over correlations in one of the plants that they looked at. So the conclusions and results of this paper, the researchers found that genetic crossing over and cytological crossing over are always accompanied by each other and have correlation. So this means that when the chromosomes are exchanging segments, they are also exchanging their genetic material as well.
Okay, and so for our selected paper, we chose an article titled Causes and Consequences of Crossing Over Evidence via a High-Resolution Recombinational Landscape of the Honeybee. So, the causes and consequences of this. The researchers utilized honeybees as their model organism to demonstrate crossing over. They used high-resolution recombination on 55 bees total from 3 different colonies. These colonies consisted of worker bees, which are the females, and these do not breed, and the queen bee, which there's only one of in each hive, who creates all of the offspring for the hive, and then the drone bees, which are male, and these do reproduce. So due to the heterozygosity in the drones, there's an interference with recombination. So when the drone and the diploid queen mate, during the meiotic recombination, they exchange homologous chromosomes and produce new combinations of alleles. The recombination of the bees resulted in GC content, which is guanine cytosine, which occurs in the nitrogenous bases. And it also resulted in increased diversity in the nucleotides and increased gene density. So the recombination rate in honeybees results in a very uneven distribution among the chromosomes. And as a result of this, the behavior of the worker bees is significantly more diverse, and the crossing over increases with the genes of the honeybees. So compare and contrast both of our papers. We found a decent number of similarities, starting with that the paper on corn and the paper on the honeybees both experienced areas of increased gene density after recombination. So for example, in our study on corn, we talked about how the areas of high density exhibited the knobs at the end of the chromosomes. And another similarity we found was that regarding both corn and bees, linkage and recombination results in increased genetic diversity. So referring back to the bees, their diversity increased regarding their behavior. Oh, I didn't mean to do that. So another similarity is that the chromosome length is impacted by the crossover rate in both bees and corn, and both also exhibit low non-crossover rates and high crossover rates, which is ideal since crossing over is beneficial to both of the organisms. And our differences is that the experiment on corn primarily focused on what happens during crossing over, and it didn't really focus on the result of crossing over, like what happened with the bees. And the study with the bees really focused on how it impacted the bees overall instead of focusing on just the chromosomes like the corn. And the last difference that we found was that the paper on the corn was aiming to see more of the physical traits of the corn, and the second paper was focusing on more of the behavioral effects. And for example, the bees exhibited varying CO2 rates, which ended up varying their recombination rates. and this impacted their overall brain expression. And those are our references. Thank you for tuning in.
Thank you.